My name is Grabrielle Brumfield. I am from Chadron State College, and I'm going to talk about some work I did in collaboration with the University of Nebraska Medical Center on platelet-mediated rescue of pancreatic cancer cells from Anchorage-Independent program cell death. Pancreatic cancer is an especially deadly disease with a relative 5-year survival rate of less than 10%. There are a lot of reasons why the disease is so deadly, one of them being that about half of pancreatic cancer patients have metastases at diagnosis. And in order for metastases to form, these cancer cells need to be able to survive while they're in circulation in transit to their secondary location. And there's a type of programmed cell death called anoicus, which is caused by lack of or improper extracellular matrix attachment. And somehow these metastasizing cancer cells are surviving that. So the question becomes, what is different about these cancer cells? Why are they able to survive? And even, is there something different about these primary cancer cells and these circulating cancer cells? So to look into that, I used some gene expression data from a study where pancreatic cancer patients had both primary tumor samples taken and circulating cancer cells taken from blood samples. And this is a heat map showing the gene expression of those circulating cancer cells, the gene expression of those in relation to the primary tumor cells. So each column is a patient, with the middle column being the average of the gene expression of each patient, and each row is a gene. And overall, over 9,000 gene probes were found to be differentially regulated between the circulating cancer cells and the primary tumor cells. So there's something different about these circulating cancer cells, and one of those differences may be leading to the survival of anoicus. I wanted to look into a little bit more of what those differences are between the circulating cancer cells and the primary tumor cells. And one of the interesting gene clusters that I found to be differentially regulated were gene clusters that we typically associate with platelets. And I thought there were probably two explanations for this. Either something about metastasis is making these circulating cancer cells express genes that we typically associate with platelets, or platelets are simply being co-isolated with the circulating cancer cells. I thought platelet co-isolation was a little bit more likely, so that's what I looked into, and I wanted to look into it as a possible mechanism of survival of these cancer cells. So my hypothesis is that interaction of platelets with pancreatic cancer cells could be helping survive this Anchorage-Independent program cell death. To do this, I used two different cell lines, T3M4 and COLO357. Both of those are pancreatic cancer cell lines, and those were either incubated by themselves or with platelets. These platelets are human platelets that were taken from the UNMC blood bank, and they were incubated in several different concentrations, most of them being about 150 platelets per mil, which is on the lower end of physiologic range. These cancer cells were either incubated by themselves or with the platelets in a low attachment plate. So these are specially coated plates that do not allow the cells to make attachments. And they were also incubated in methylcellulose media, which is a thicker type of media, so that these cells aren't sinking to the bottom or clumping as normally they might be able to in normal media. These cells were then isolated at different time points, and they were then investigated using MTT, flow cytometry, Western blotting, and microscopy. So I wanted to look into whether or not there was a physical interaction between platelets and cancer cells that might make them be isolated together. This was done using the COLO357 and T3M4 cell lines, and these cells were either untreated with no platelets or treated with platelets on the right with 150 platelets per mil. And then at 0 and 12 hours, pictures were taken at 200x. At both of the untreated groups for each of the cell lines, there isn't a lot of clumping in the cells. And in addition to the platelet groups, there also doesn't seem to be much clumping. And then at 12 hours, in both of the untreated groups, there seems to be a little bit of clumping. However, we see much more in both of the platelet-treated groups at 12 hours. You can see, especially at the areas denoted by the red arrows, that there does seem to be a lot of interaction with those larger cancer cells with those smaller pancreatic cancer, or those smaller platelets. So from this, it does appear that platelets and these cancer cells are physically interacting. To further validate that, I used some flow cytometry with fluorescently stained platelets and unstained cancer cells. With these platelets alone, the y-axis being side scatter and the x being forward, we see that these platelets, being relatively small, are in this gate, with also showing that they have a low granularity as they are activated. Then if we look at this gate to the right, this is the gate where we would expect to see any fluorescently stained cancer cells, as the cancer cells are much larger, and but we don't really see anything in there, as the cancer cells are, as I said, much larger, so the platelets don't enter into that gate. Then as we look at the cancer cells alone, with the x-axis being the forward scatter again and y being side, the cancer cells are showing up as being much larger with a larger granularity, as we would expect, with only some background showing in the platelet gate. And then we see very little background into that gate where we would expect to see any fluorescently stained cancer cells. But as I mentioned, I did not actually stain any of these cancer cells. However, and then adding platelets that are fluorescently stained, there is an increase in that gate where, as I said, this is where we would expect to see any fluorescently stained cancer cells. But these cancer cells are not stained, so that physical interaction between those platelets and cancer cells do appear to be making these cancer cells appear to be fluorescent here. This is a graphical representation of that flow cytometry data, with on the y-axis the percent CFSE stained positive events in that gate, and then the x-axis being the treatment groups, with an increase in platelet treatment on the bottom. And as we see an increase in that fluorescently stained platelet treatment, there's an increase in those percent CFSE positive events. So from this, we do infer that these platelets and cancer cells are physically interacting. Next, I wanted to see if these platelets are actually giving a survival benefit to these cancer cells. And to do that, I wanted to first validate that these cancer cells do die in these Anchorage-independent conditions. Using the T3 and 4 cell line and a viability stain, I incubated these T3 and 4 cells in Anchorage-independent conditions from 0 hours to 24 hours with the dead cell stain intensity on the y-axis and forward scatter on the x. And we see here that at 0 hours, about 85% of cells are standing as alive. And as the time increases at 24 hours, only about 30% of these Anchorage-independent incubated cancer cells are alive. So from this, we infer that these cancer cells do die from these conditions. Next, I wanted to see if these platelets would rescue these cancer cells from this programmed cell death. And with incubating these cells at 150 million platelets per mil, the T3 and 4 and Colo-37 cell lines, at 12 hours we did see a statistical difference in survival of these T3 and 4 cell lines. However, this is on the lower end of the physiologic normal for platelets. So I wanted to look more into if there would be a difference with an increased platelet treatment. And with the T3 and 4 cell lines at both 12 and 24 hours, we did see many statistical differences in survival of these untreated versus platelet-treated groups. However, I did not see the same trends in these Colo-37 cell lines. I'm not sure about why there wasn't that same trend we saw in the T3 and 4, but I wanted to further explore those T3 and 4 cells. And to do that, I first turned back to bioinformatics. And first, I looked into types of genes that were differentially regulated between those circulating cancer cells and the primary tumor cells, as well as any hematologic cells. So I wanted to look into genes that were being attributed only to these circulating cancer cells. And one thing that I found is that many G protein coupled receptor signaling genes were found to be differentially upregulated. And notice that this x-axis is the negative log of the p-value of this significance. So these are very significantly differentially regulated. So I used Western blotting to look if there were any differences in G protein coupled receptor signaling related proteins in these platelet treated cells using the T3 and 4 cell line. And notice that I do have a beta actin control on the bottom, as well as a platelet control. This platelet control was a well that was just loaded with the same volume of platelets that I used to treat these cells. And a CD42C platelet marker was used to look for any presence of platelets in these samples. And we do see a large amount in the platelet control, as expected, but we see little to none in any other well, which suggests that any protein differences we see here are coming from those cancer cells alone and not residue from those platelets. We do see that there does appear to be a slight difference with a slight increase in phospho-ERK expression in these platelet-treated cells, which is associated with an increased G protein coupled receptor signaling activity. If we saw an increase in phosphorylated SARC, that would also be associated with an increase in G protein coupled receptor signaling. However, I did not see that increase. The reason for that is still to be explored. But in conclusion, from this presentation, platelets and cancer cells do appear to be physically interacting in Anchorage-Independent conditions, and it does appear that platelets are offering a survival benefit to these pancreatic cancer cells, and that survival benefit could be tentatively through increased G protein-coupled receptor signaling. I'd like to thank the Batra Lab, specifically Andrew Cannon, Dr. Sushil Kumar, and Dr. Batra, and this work was supported by the INBRE program with the grant number listed here. Here are my resources, and thank you for listening.